Hello everyone, welcome to this extended session related to microbiology and biotechnology. Myself Mori Dawal, Assistant Professor at BK Modi Government Pharmacy College. So if you remember last time when we were discussing about the questions related to microbiology and biotechnology, one of the last questions that we have discussed was about hybridoma technology, and the question was these hybrid cells are selected by using which media. So because in that lecture, due to time constraint, I was not able to explain the mechanism by which these cells are selected. So I have decided to make a separate video where I am going to explain that how HAT media works and how it is helpful in selecting the hybridoma or the hybrid cells. So basically, if we see the basic mechanism, what is hybridoma cell? Basically, it's a fusion cell which is formed by combining the spleen cell, or it is also known as a beta cell, with myeloma cell. So here basically these spleen cells are they are beta cell which are responsible for producing antibodies, and myeloma cell they have the ability to divide rapidly, and that is it has the ability to divide rapidly, and whether when these two cells are fused, whether the fusion has been complete or not, that is checked by growing this cell in a specialized media that is known as a HAT media. So how this HAT media works, that is the basically theme of this extended session. So we know, or you might be knowing that any cell when it divides, first thing that required to do is to synthesize DNA, because every new daughter cell they require to have a same set of the DNA that is in the parent cell. So for the cell division, the first condition is that DNA required to be synthesized. So generally there are two pathways by which DNA can be synthesized. When we say DNA, in a broad term, what is DNA basically? We know it's a deoxyribose nucleic acid, sugar, phosphate, and 4 nitrogen base pair: adenine, guanine, thymine, and cytosine. So when we say the need is to synthesize DNA, basically what we require to synthesize these 4 nitrogen base along with sugar and the phosphate, and that is also known as something assembly that is called as a nucleotide, right? So we require nucleotide. So DNA can be synthesized basically by two pathway. One is known as a de novo pathway, and second is known as a salvage pathway. What is de novo? De novo is something if it is synthesized by using very simple molecules from start to end. If the starting is very simple molecule, then that kind of the synthesis is known as a de novo synthesis, new synthesis. So let us see DNA. In DNA, if we categorize the nitrogen base, it is basically 2 nitrogen bases are there: purine and pyrimidine. So let us see how these 4 nitrogen bases are synthesized by first de novo pathway. So as I said, de novo is where the starting material is a simple molecule and then end product is the purine or pyrimidine base pair. So if we see this purine biosynthesis, purine nitrogen base pair, that is basically adenine and guanine. So the starting point is ribose 5-phosphate, and if you see this entire biosynthetic pathway, we are not going to discuss all of the steps. What we are going to discuss, which are important to understand the functioning of HAD media. So as you see here, the starting material is ribose 5-phosphate, and by series of enzymatic reaction, it gets converted into a intermediate that is known as a eosine monophosphate. So eosine monophosphate is a very important it's a very important molecule which further can be converted into required nitrogen base pair. So you can say eosine monophosphate is an intermediate. So once eosine monophosphate has been synthesized, this eosine monophosphate, after the subsequent reaction, it get converted into, as you see, eosine monophosphate. Due to further enzymatic reaction, it gets converted into ATP, that is adenine triphosphate, and guanine triphosphate. So it's a nucleotide. So two nucleotide, purine based nucleotide, adenine and guanine, are synthesized. Its starting material is ribose 5-phosphate, intermediate is eosine monophosphate, and after the further reaction it's converted into the adenine triphosphate and the guanine triphosphate. So that is the two nucleotide that we are looking for. Same way, if we talk about the other nucleotide, so now we have adenine and guanine, and as you see, this starting material here is a simple molecule, and that is why it is known as a de novo synthesis. So next de novo synthesis, let us look for the pyrimidine. So in a pyrimidine, as you can see here, the starting material is carbon dioxide and glutamine, and as you see, after the series of enzymatic reaction, it converted into uridine diphosphate, that is an intermediate, and this intermediate after the subsequent reaction converted into thiamine monophosphate and cytosine triphosphate. So this you can say thiamine and cytosine, it's a direct nucleotide, it's a monophosphate. So generally this thiamine monophosphate, when it react with the enzyme thiamine kinase, it convert into thiamine monophosphate. When it react with the thiamine kinase, which is a enzyme, it convert into thiamine diphosphate and it convert into thiamine triphosphate. So triphosphate, we know it's a nucleotide. So we know what is nucleotide: phosphate, sugar and nitrogen base. So by this pathway we have pyrimidine base pair, cytosine and thiamine. Now if you observe this reaction, in both the reaction where we have discussed purine and pyrimidine, if you see one step, let me show you. Okay, so if you see in both these synthesis, one step that is required to be concentrated to understand the mechanism of HAT media is something called here. So here if you see the reaction, one of the intermediate reaction is that a formyl folate is converted into folate. So here you can see in this reaction, it's one of the intermediate step where we require formyl folate to be converted into folate, and then the reaction is proceeding. Same way, if you observe the reaction in the pyrimidine biosynthesis also, in the pyrimidine biosynthesis also, here also we find there is a one step where this same reaction is proceeding. So here also if you see in the synthesis of thiamine monophosphate, here methylene tetrahydrofolate is converted into tetrahydrofolate. What is tetrahydrofolate and this methylene tetrahydrofolate? I am just discussing subsequently, but just understand over here that there are two steps where we are finding that folate molecules are participating. So this formyl folate is converted into folate, and that reaction required to complete to make sure that we have eosine monophosphate, which subsequently converted into adenine and guanine. And same way here to make sure that we have the thiamine, here also we required a methylene tetrahydrofolate as a intermediate, so that this reaction can be completed. Now see what is basically tetrahydrofolate dihydrofolate. So basically these are formed from the folic acid. So these are the derivatives of the folic acid. So folic acid is known as it's a food supplement. In body it is converted first into dihydrofolate. This dihydrofolate it convert into tetrahydrofolate. Now the reagent or the enzyme which is responsible for converting dihydrofolate into tetrahydrofolate is known as dihydrofolate reductase, and that convert into tetrahydrofolate. Now this tetrahydrofolate after the subsequent reaction converted into 10-formyl tetrahydrofolate, and as we have seen over here, this is the one of the reagent that we required for completion of the synthesis. So formyl tetrahydrofolate it's convert into folate. So here that is we required over here for the purine synthesis, and same for the pyrimidine synthesis. As you see, methylene tetrahydrofolate it again converted into tetrahydrofolate. So here as you can see that these two reagents are required to make sure that three nitrogen base pair adenine, guanine and thiamine are synthesized. Now let us see how both the reactions are happening simultaneously and how it is used for the selection. Let us say, so if you see over here, that this is known as a de novo pathway. Now, by case, if the cell is not able to use this de novo pathway to synthesize the required nucleotide, a second pathway is also available, that is known as a salvage pathway. What is salvage pathway? Salvage means rescue, right? So this is the optional pathway. Whenever there is a first priority, cell is always going to synthesize by de novo pathway. But whenever by anyhow, if cell is not able to follow this de novo synthetic pathway, then a second optional pathway is there, that is known as a salvage pathway. What is the salvage pathway? Here what cells are doing, they are taking directly food supplement and they are directly converted into respected nucleotide. So let us see example. So when this de novo synthesis is not working, and suppose a cell has to make purine nitrogen base pair, what they can do, they can take this hypoxanthine, which is one of the food supplement. So they take hypoxanthine, and this hypoxanthine, when they react with hypoxanthine guanine phosphoribosyl transferase enzyme, it converted into eosine monophosphate. And if you remember the reaction that we have, once you have the eosine monophosphate, subsequently you can produce adenine and guanine. So purine nitrogen base pair, the salvage pathway start with the hypoxanthine, and it is catalyzed by enzyme that is known as a hypoxanthine guanine phosphoribosyl transferase. Same way, if you see basically what is happening in the myeloma cell. So whenever we are producing this hybridoma cell, generally the selection is something like that. The spleen cell or the beta cell which are selected, they require to have this SGPRT plus. That means they require to have a ability to synthesize this hypoxanthine phosphoribosyl transferase, and that is why these cells are known as SGPRT plus. That means this cell required to have a ability to synthesize this enzyme which can react or which can convert hypoxanthine into eosine monophosphate, and subsequently you can say into purine. And to contrary to that, to differentiate between them, other part of the cell, other part of the hybridoma cell, that is the myeloma cell, they require to have SGPRT negative. That means these cells, they don't require to have ability to synthesize SGPRT enzyme on their own. So here one cell can produce, that is the beta cell can produce this enzyme which has the ability to convert hypoxanthine into eosine monophosphate and subsequently purine, and here they don't require to produce this one. So now basically you see what we required for the DNA synthesis. So for the DNA synthesis we required 4 nitrogen base pair, right? Adenine, guanine, thymine and cytosine. So what is the how this pathway is working? So first requirement, as you see over here, characteristic is that myeloma cell, which is the rapidly dividing cell, it is not having SGPRT. And because it is not having SGPRT, you can say single myeloma cell, if by chance they are not following de novo synthetic pathway. As we discussed, de novo, it's the main pathway for synthesis of the DNA. But because now these cells are SGPRT negative, they cannot go for salvage pathway. So for them, DNA division is, or the synthesis of the DNA is completely blocked. Now spleen cell, which are producing the antibodies, these cells are required to have their ability to produce SGPRT. So here you can understand that what is happening with this spleen cell or beta cell, that by chance if they don't have ability to follow the de novo synthetic pathway, they can have an optional pathway available, that is the salvage pathway, because they have this enzyme, they can use this enzyme and they can have an alternate pathway to synthesize the purine base pair. So what is basically, now discuss what is basically HAT media. So as you know what is HAT, it stood for hypoxanthine, aminopterin and thiamidine medium. So it is having 3 components. First, hypoxanthine, aminopterin and thiamidine. So let us see one by one how each ingredient is applicable. So first let us see the most important part is aminopterin. What is aminopterin? So aminopterin is a basically it is a competing molecule which compete with the dihydrofolate. So let us see how it's happening. So if you remember the mechanism that I have explained, that folic acid that is convert into dihydrofolate. Now this dihydrofolate, when it react with the dihydrofolate reductase, it convert into tetrahydrofolate. And again we go through this one. We know once tetrahydrofolate we have, it convert into formyl tetrahydrofolate and methylene tetrahydrofolate. And both are required to complete the synthesis of here thiamine, or you required for the complete the synthesis of adenine and guanine. So what this aminopterin does, it compete with this enzyme, it compete with this dihydrofolate to join with dihydrofolate reductase. So basically what this aminopterin is doing, ideally when this enzyme is available, this enzyme should bind with the dihydrofolate and it should convert dihydrofolate into tetrahydrofolate. So this tetrahydrofolate subsequently can convert into methylene tetrahydrofolate or formylene tetrahydrofolate, and they can complete the de novo synthetic pathway. But here, because this aminopterin, it's competing with the space, it's competing with this dihydrofolate, what aminopterin will do, it will bind with this enzyme. And once it binds with this enzyme, so this enzyme no longer can convert dihydrofolate into tetrahydrofolate. And as you can expect, once we don't have the tetrahydrofolate, this is going to stop the pathway for synthesizing 3 nitrogen base pair. So let us go back to so let us see again. So if we don't have the tetrahydrofolate, if you don't have this formyl tetrahydrofolate, then you don't have this reaction to be completed. So you don't have the eosine monophosphate, and eosine monophosphate you don't have adenine and guanine. Same way, if you don't have this methylene tetrahydrofolate, you don't have thiamidine. So what basically this aminopterin is doing, it is inhibiting the synthesis of adenine, guanine and thiamine by de novo synthesis pathway. Remember, it's a de novo synthesis pathway which they are inhibiting. So now, because this de novo synthesis pathway is not there to produce adenine, guanine, thiamine and cytosine, obviously the cell required to find alternative ways. And what is the alternative way? Alternative way is the salvage pathway. But here also, what is the condition over here? So as you see, to have the salvage pathway, what is the required? First thing required is that we require this enzyme, right? Hypoxanthine guanine phosphoribosyl transferase, which can convert this hypoxanthine into eosine monophosphate, and which subsequently convert into adenine and guanine. So if cell don't have this, then they can no longer follow the salvage pathway. And remember what we said, myeloma cells are selected in such a way that they don't have SGPRT. So because they don't have SGPRT, they can no longer synthesize the DNA by de novo synthesis. Why? Because already aminopterin will not allow them to synthesize, and because they don't have a SGPRT, they cannot synthesize salvage pathway. So basically what happens when we fuse the cell, two cells, suppose this is spleen cell and this is myeloma cell. So there are 4 possibilities can have. One possibility is that 2 spleen cells are joined. Second possibility is that 2 myeloma cells are joined. Or third probability is that a spleen cell joined with myeloma cell, and that is what we call hybrid, or a proper hybrid cell. It's a complete fusion. So first scenario, suppose when 2 spleen cells are joined, what happens, or 2 beta cells are joined. When 2 beta cells are joined, what is the major problem with this 2 beta cells? They are having very short half-life. So even if they join, they are not going to survive for longer duration. So when you put it in the media, HAT media to grow, they are not going to grow because their de novo synthetic pathway is, and they have their short life, so they are not going to work. Second, when 2 myeloma cells are joined and when they are growing, because myeloma cell has the ability to divide very rapidly. So they can divide. But what is problem with the myeloma cell? That first, because our HAT media is containing aminopterin, it will allow this cell to not follow the de novo synthetic pathway. It will not allow the cells to have a de novo synthetic path. And second thing, that because this myeloma cell don't have the SGPRT, hypoxanthine guanine phosphoribosyl transferase enzyme, they can no longer go for salvage pathway also. So ultimately, they don't have a way to go to de novo pathway or a salvage pathway. They cannot synthesize new DNA and ultimately they will die. So only cell where there is a one spleen cell or a beta cell which is having SGPRT, so it can produce SGPRT, and it is also having myeloma cell. So myeloma cell will divide rapidly, so cell will divide, and whenever they divide, whatever DNA they require to synthesize, these cells are going to synthesize by using salvage pathway. So only spleen cell, beta cell, it's not going to survive because it is having the short lifespan. Only myeloma cell not survive because they cannot follow either de novo or either the salvage pathway. The only cell which has the ability to survive is the cell which is having the proper fusion, that is having beta cell as well as having the myeloma cell. They are only going to divide. So what happened that when you use HAT medium, so aminopterin, it's compete with this dihydrofolate detectase, and this reaction is not complete, and that is why synthesis of adenine, guanine and thiamine is reduced. So obviously to have the salvage pathway, see spleen cell, the beta cell has SGPRT. So what they are going to do, in the media, HAT media, another component is hypoxanthine, and as we have discussed, this hypoxanthine it react with the SGPRT and it convert into eosine monophosphate, and as we have seen, eosine monophosphate is the intermediate which subsequently get converted into purine base pair, that is adenine and guanine. So you have complete adenine and guanine. So now purine base pair is where? What is the another base pair we required? Is the another base pair that we required is the pyrimidine. So the another component of this media, that is the thiamidine. So hypoxanthine, aminopterin, and last one is thiamidine. So thiamidine, it react with the thiamidine kinase. It's an enzyme. It convert thiamidine into thiamidine monophosphate. Then it convert into thiamidine diphosphate and thiamidine triphosphate. That is the nucleotide. So we have adenine and guanine, which is coming from hypoxanthine, and we have the thiamine, which is coming from thiamidine. So if you observe this one, this reaction, we have only synthesized, or we have only able to synthesize 3 nucleotide base pair. What about cytosine? So for cytosine, you see, for the cytosine, the point is that to synthesize the cytosine, there is no role of dihydrofolate or tetrahydrofolate. So again, if you observe this reaction is required for eosine monophosphate, and eosine monophosphate is the intermediate that is required for both adenine and guanine. Same way, if you see for pyrimidine, uridine diphosphate is the intermediate, but if you see only to synthesize thiamidine, we required methylene triphosphate, methylene tetrahydrofolate. To synthesize cytosine, there is no function of tetrahydrofolate or dihydrofolate, and that is why cytosine pathway is not going to inhibited. Only pathway which aminopterin can only inhibit the synthesis of thiamidine, adenine and guanine, and that is why if you see the media that we are using, these media are supplementing the requirement for hypoxanthine, ultimately adenine and guanine, and thiamidine, that is not for cytosine. So that is how this HAT media works. Aminopterin, it blocks the synthesis of adenine, guanine and thiamine by inhibiting the de novo synthesis, and hypoxanthine and thiamidine, they start with the salvage pathway. So by salvage pathway, these are converted into supplementing the requirement of adenine, guanine and thiamine. So that's about this. Thank you everyone for patient listening. So as you know, it's a little bit of complicated mechanism with HAT media, and because in a last lecture we have the time constraint, I was not able to explain, so I decided to make this another video for this one. Thank you everyone. Please share your valuable feedback on it. Please say if anything we can improve. We need to add something, or we need to restrain ourselves from discussing further. So please share your feedback. Thank you very much.